The human genome contains 3 billion letters of biological code. The ultimate challenge is locating one specific error hidden somewhere in that massive sequence. To solve a scale problem this large, researchers rely on a biological and mathematical tool known as a Genome-Wide Association Study, or GWAS. Without it, looking for the root cause of a trait is like searching an entire continent for a specific house without a map. Traditionally, geneticists map traits using quantitative trait loci, or QTL mapping. This involves tracing diseases through specific family trees or controlled laboratory crosses. Because a pedigree only covers a few generations of reproduction, the parents' chromosomes haven't had much time to mix and shuffle through meiotic crossover. As a result, massive chunks of their DNA are inherited exactly as they were. This creates a major bottleneck. These large chromosomal blocks keep hundreds of distinct genes trapped together in a single package, resulting in terrible mapping resolution. If you want to cure a complex, multifactorial disease, neighborhood-level resolution is useless. Researchers require single nucleotide precision. The first step in a GWAS pipeline is to abandon the family tree entirely. Instead, we sample huge global populations of completely unrelated individuals. While these individuals don't share immediate parents, they do share a distant evolutionary ancestor. Their genomes have been mixing for thousands of generations. All that historical recombination acts like a biological blender. It takes those large, clunky chromosomal blocks from our ancestors and slices them into incredibly tiny, isolated fragments. Tapping into deep evolutionary time is how we refine our mapping resolution from millions of base pairs down to the dimensions of a single gene. This brings us to a core genetic mechanism, linkage disequilibrium, or LD. This is the tendency for specific alleles to be inherited together simply because they are physically close on a chromosome. Over generations, recombination decays this association. The physical distance dictates the math. Larger distances between alleles allow for more frequent meiotic crossovers, slicing away those distant markers over time. Only the alleles sitting immediately next to the causal mutation manage to survive this decay. They stay glued together in high linkage disequilibrium. By tracking these persistent markers, GWAS can reliably flag the location of a disease-causing variant, even if we haven't sequenced the mutation itself. The proxies we track are called Single Nucleotide Polymorphisms, or SNPs. These are single-letter variations in the DNA sequence. Researchers analyze millions of them simultaneously. This animation shows the statistical trap waiting in large-scale genomics. If you test 1 million independent SNPs against a standard probability threshold of 0.05, random chance dictates that roughly 50,000 false positive associations will slip right through. To fix this, researchers apply a rigorous adjustment known as the Bonferroni correction. We take that nominal threshold and divide it by the total number of tests we are running. Doing that gives us a hyper-strict genome-wide significance threshold of 5 × 10 to the -8th power. The loose filter is replaced by a heavy, restrictive barrier. Surviving this extreme mathematical gauntlet is the only way to separate an authentic genetic signal from a sea of random evolutionary noise. This chart is called a Manhattan plot. It's a specialized scatter plot we use to visualize the data points that survive our statistical filter. The horizontal axis represents the physical coordinates of the human genome, arranged sequentially from chromosome 1 down to 22. The vertical axis measures statistical significance, quantified as the negative logarithm of the p-value. That hyper-strict Bonferroni adjustment we calculated earlier forms a horizontal line right at 7.3. The highly significant genetic associations rise above it, clustering densely together like skyscrapers. Peaks rising above this threshold represent authenticated genomic loci. These signals identify the specific chromosomal regions associated with the trait, narrowing our search from millions of possibilities to a handful of high-confidence markers. By leveraging ancient population diversity, utilizing linkage disequilibrium, and applying these ruthless statistical filters, researchers can isolate the precise genetic signals linked to complex traits. GWAS transforms evolutionary history into a high-resolution map, providing the coordinates needed to unravel the biological architecture of modern disease.